Welcome back to the Deep Dive, where we take the most comprehensive, complex scientific research, distill it down, and give you the essential knowledge to be immediately well-informed.
And our mission today is arguably one of the biggest we've ever tackled.
I think so. We're confronting time itself.
Indeed. We are moving far beyond the superficial signs of aging, the wrinkles, the gray hair, to focus on the deep molecular engine failure that inevitably leads to chronic disease.
That's really the core premise, isn't it? That aging is this gradual, irreversible pathophysiological process.
Right, and it's the single greatest driver of, exponentially increased risk for virtually every major disorder you can name.
Things like Alzheimer's, Parkinson's, cardiovascular failure, metabolic collapse, arthritis, and just a general breakdown of the immune system.
All of it. Our source material today is a monumental review detailing the molecular, cellular, and systemic drivers of this decline, what are famously known as the hallmarks of aging.
So we're going to map out these drivers, show how they converge to cause disease, and then explore the incredibly challenging world of anti-aging interventions.
And the consensus among biologists is, it's pretty clear at this point, aging is the leading risk factor.
It's not just a risk factor, it's the risk factor.
Exactly. So if we can truly understand and intervene in the aging process itself, we might be able to address a whole spectrum of these diseases simultaneously, rather than, waiting for them to pop up one by one.
Before we jump into the 10 mechanisms, I think it's worth taking a quick look at the history here, because it shows how much plasticity is actually inherent in aging.
That's a crucial point. It's so easy to feel like aging is just this fixed, unchangeable fate.
But it's not. Researchers have been demonstrating external influence for nearly a century now.
It's a fascinating historical nugget. If you go back to, say, 1925, there are these surprising experiments where researchers discovered that just manipulating the intensity of light, just light, it could impact the growth rate and even the lifespan of Drosophila, the common fruit fly. It was one of the earliest, most powerful demonstrations that environmental factors could have this profound influence.
And that was long before we were thinking about genetics in the same way.
Right. And then, of course, came the famous caloric restriction studies. They showed that significantly cutting calories could impact longevity and mitigate age-related pathologies in rodents and other organisms.
But the modern era, the sort of high-resolution molecular study of aging, that's surprisingly recent.
It is. It wasn't until the discovery of the first long-lived genetic strains, specifically in the microscopic roundworm C. elegans about 30 years ago, that we realized longevity wasn't just about environment or calories.
It was genetically programmable.
Exactly. That discovery truly ushered in this era where researchers started cataloging and identifying the discrete molecular failures we're about to discuss.
So the ultimate big picture, then, is that understanding aging helps us identify therapeutic targets for things like diabetes, Alzheimer's, COPD, osteoarthritis.
You're moving past just treating the symptoms and going straight for the root cause.
That's the goal.
So how do we impose some order on this vast, incredibly complex process of system-wide decline? Biology is messy.
It is.
But back in 2013, the field established a governing framework, 9 distinct features or hallmarks that define the aging process.
And science is always moving forward. We've recently added a 10th key feature, which we'll get into, compromised autophagy.
Right. So these 10 hallmarks provide a kind of standardized mechanistic map for every researcher looking at age-related decline. It gives us a common vocabulary for this failure.
To organize this for everyone listening, we can categorize these 10 hallmarks into 3 distinct levels of mechanism.
We can, yeah. We're going from the microscopic failures inside the nucleus all the way up to system-wide breakdowns in communication and sensing.
So let's lay out that map. We'll start with 6 hallmarks at the fundamental molecular level.
That's genomic instability, telomere dysfunction, epigenetic alterations, loss of proteostasis, that new one compromise of autophagy, and finally, mitochondrial dysfunction.
Okay, that's the molecular layer. Then we move up to 3 hallmarks operating at the cellular level, where all these damaged components start to interact.
And those are cellular senescence, stem cell exhaustion, and altered intercellular communication.
And finally, there's one key systemic alteration that affects the whole organism's ability to respond to its environment,
which is deregulated nutrient sensing.
Yeah.
It's a structure that helps us organize this just tremendous amount of data.
So let's start small. Let's look at those core molecular mechanisms, beginning with the integrity of the cell's blueprint, the genome.
Genomic instability is, to put it simply, it's the accumulation of damage to your DNA over time.
Our DNA is under constant assault, right? From both internal and external forces.
Constant. And this leads to molecular lesions, point mutations, and most critically, DNA double-strand breaks. This is a primary quantifiable marker of that instability.
And when the cell detects one of these double-strand breaks, it basically goes into emergency mode.
Total emergency mode. It activates the DNA damage response using these highly complex pathways like p53, p21, and p16INK4A PRB.
And the cell's first priority isn't even repair, is it? It's just survival.
Survival first. It hits the brakes, blocks the cell cycle, and makes sure that damaged information can't be copied and passed on.
What's truly eye-opening to me is the sheer universality of this damage. I mean, it's everywhere.
Absolutely. Researchers use a tool called the tabula mira senescence, which is this comprehensive single-cell atlas of aging mice, to map this process across over 350,000 cells.
And they confirmed, maybe not surprisingly, that genomic mutations increase with age in all the organs they studied.
But here's the surprise you mentioned.
Yeah, the surprise is which organs were most affected. It wasn't the brain or the heart, which you'd think of first.
Right. Their analysis showed the tongue and the bladder were the most significantly affected.
Which just demonstrates that aging isn't only hitting the high-profile systems, is a full-body breakdown, often in unexpected places, maybe due to high cell turnover or constant exposure to the environment.
Now, here's where this very basic damage mechanism spirals into a system-wide inflammatory problem.
This is a critical link.
When that nuclear DNA damage becomes chronic, it leads to the formation of these persistent structures that are called DNA scars. DNA segments with chromatin alterations reinforcing senescence.
So the damage becomes a permanent fixture in the cell.
It does, and that permanent damage leads to a loss of structural integrity in the nucleus. We see a loss of a key nuclear membrane protein called lamin B1 in these senescent cells.
So the protective boundary of the nucleus starts to weaken?
It weakens, and when that happens, pieces of chromatin, what we call cytoplasmic chromatin fragments, or CCF, they escape the nucleus and enter the cytoplasm.
This is a critical failure point.
It's huge,
Sure.
because when the cell sees its own damaged blueprint fragments floating loose outside the nucleus, it basically mistakes them for a viral invasion.
Okay, let me make sure I've got this analogy for you. The nucleus is the vault with the blueprint. Lamin B1 is the security guard.
Right.
The guard fails, the blueprint leaks out, and the rest of the cell, which is constantly scanning for invaders, sees this internal DNA as foreign and triggers the ultimate inflammatory alarm.
That's a perfect analogy. The CCF is recognized by a sensor called CGAS, which then activates the S-sting pathway.
And S-sting is a major mediator of the inflammatory response.
It is. It drives this critical component known as the senescence-associated secretory phenotype, or SAP. So the failure of genomic integrity isn't just internal damage anymore. It's now actively driving chronic low-grade inflammation throughout the entire organism.
It's the sound of the house burning down, driven by its own broken security system. Now, we also have to talk about mitochondrial DNA, or mtDNA. Why is this genetic material so uniquely vulnerable?
Well, because the mitochondria are the cell's powerhouses, they are a high-energy environment. This means they're a major source of reactive oxygen species, or ROS, basically molecular exhaust.
And unlike the nuclear DNA, mtDNA doesn't have the same protection.
Exactly. It lacks the histone proteins that cushion and protect the nuclear blueprint, and it has a much less effective repair system.
So ROS generated right there in the powerhouse is causing damage to the DNA that controls the powerhouse itself. It's a direct self-destructive cycle.
It is. And we see decreased mtDNA copy numbers and elevated mutation rates in aging organisms, which creates this catastrophic functional decline in energy production.
And let me guess, this damaged mtDNA can also leak out.
It can. Damaged mtDNA can also be released into the cytoplasm, where it binds to CGAS and activates the S-sting pathway, just like the nuclear fragments.
So this powerfully links mitochondrial decay and genomic instability directly back to that same systemic inflammatory SISP response.
It all connects.
Okay, so if genomic instability is the accumulated structural damage, telomere dysfunction is often seen as the ticking time clock.
That's the classic analogy. Telomeres are these highly conserved repeat sequences, TTDGs in mammals, at the ends of our chromosomes. They act like the plastic tips on your shoelaces, just maintaining genomic stability.
And they shorten with every single cell division.
They do, until they reach a critical point known as the Hayflick limit, and the cell interprets that exposed and as a DNA double-strand break.
Which triggers the same DNA damage response we just talked about, stopping the cell cycle and inducing senescence.
Right. The natural maintenance for these tips is an enzyme called telomerase. Its activity is highest in embryonic cells, and then it progressively decreases with age.
Which is thought to be a way to suppress cancer risk, right?
It is. But this leads to the fundamental therapeutic paradox that defines this whole area of research.
The double-edged sword.
A very sharp one. On one hand, telomerase reactivation is highly promising. Overexpressing it successfully mitigated aging effects and prolonged lifespan in mouse models of progeria. That's a condition of accelerated aging. It literally reverses the clock.
And this is a huge but.
The challenge is that telomerase overexpression is found in approximately 90% of all human cancers. It's the mechanism cancer cells hijack to achieve immortality and just keep proliferating.
So any therapeutic use has to be navigated with extreme caution. How do you turn it on just enough to repair tissue, but then ensure it turns completely off so you don't promote a tumor?
That is the high-stakes engineering problem. It's a balance between immortality and malignancy.
Okay, moving beyond the DNA sequence itself, let's look at the epigenetic alterations. This concept refers to changes in what the cell does, the gene expression, that aren't caused by changes in the DNA sequence.
Right, we're talking about everything surrounding the DNA methylation, histone modifications, and the whole ecosystem of non-coding RNAs.
DNA methylation is like the cell's dimmer switch, right?
Perfect way to put it. It's adding a methyl group to a cytosine, which typically silences a gene. It dictates which genes are accessible and which are locked away.
And with age, we see what researchers call epigenetic drift.
We do. The pattern of methylation changes drastically. You get a general decrease in methylation in some parts of the genome, but paradoxically, you get hypermethylation, increased silencing in the promoter regions of other key genes.
So it's a loss of precise, coordinated control.
It is, and these shifting patterns are so predictable and so reliable that they actually gave rise to the concept of the epigenetic clock.
This is the idea many of our listeners have probably heard of. The Horvath clock from 2013 uses these specific methylation patterns to measure our biological age.
And it can predict lifespan, often more accurately than just your chronological age. Later versions, like the D&M phenoage, refine this even further.
And confirm that the speed of this clock is intrinsically linked to these fundamental molecular aging processes, especially inflammation and DNA damage.
These clocks prove to be incredibly sensitive measures of health. We know, for instance, that the biological clocks of people with fatty liver disease move significantly more quickly.
They are aging faster than their birth certificate would suggest.
While on the other hand, you have centenarians, people who live to be 100 or more, and their clocks often move more slowly throughout their lives. It's powerful evidence that some people are just aging more slowly at a molecular level.
And that's such a profound insight for you, the listener. Lifestyle changes have measurable, quantifiable impacts right down at this level. Improved diets have been shown to slow down certain biological clocks.
Which means things like chronic stress or poor sleep. They're probably speeding it up. The impact is real and measurable.
Beyond methylation, there are histone modifications. Histones are the proteins that DNA wraps around, like thread on a spool.
And modifications to those spools, like acetylation and methylation, determine how tightly the DNA is packaged. These also shift drastically with age. In models of premature aging syndromes, we see this widespread loss of heterochromatin, the tightly packed, silenced DNA.
So it's a loss of structure, a relaxing of the whole organization system.
Right. And this structural failure is deeply regulated by cellular energy, which connects it directly back to the mitochondria.
How so?
Well, when cells experience mitochondrial stress, the tricarboxylic acid cycle gets impaired, and that reduces the production of something called acetyl-CoA.
And acetyl-CoA is necessary for histone acetylation.
Precisely. So less acetyl-CoA means less histone acetylation, which causes chromatin to reorganize and lose its structure. The fuel of the powerhouse literally affects the structure of the blueprint.
It's also interconnected. And the last piece here is transcriptional alterations involving non-coding RNAs.
These are regulatory molecules that don't code for proteins but control gene expression. MicroRNAs, for example, secreted by certain stem cells, decrease during aging.
But you can add them back?
You can. External treatment with exosomes, these tiny vesicles containing those miRNAs from healthy cells, can actually slow the aging rate in recipients.
That is incredible.
Then you have things like circular RNAs, which are enhanced during aging in the brain, probably because their circular structure makes them super stable. They just hang around causing trouble.
And what about that junk DNA?
The accumulation of double-stranded RNA from those repetitive junk elements. That activates the innate immune response and drives the neuroinflammation we see in almost all age-related neurodegenerative diseases. It's yet another way internal molecular waste triggers an external inflammatory catastrophe.
So now we get to the core cellular maintenance systems, starting with the loss of proteostasis. This is the inability to maintain protein quality control.
This is where the cell's machinery really starts to rust. And it's a major factor in aging and nearly all of those protein misfolding diseases like Alzheimer's and Huntington's.
The cell has these internal quality control teams, right?
It does. You have chaperones or heat shock proteins that help misfolded proteins fold correctly. And you have degradation systems, the proteasome for short-lived proteins, and the autophagy system for bigger structures.
And when any of those fail, toxic protein aggregates just build up.
They build up fast. And there's this constant tension in the cell between growth and maintenance. This is perfectly embodied by insulin-like growth factor 1, or IGF-1.
Right. Inhibiting the IGF-1 pathway is known to increase lifespan in simple organisms like worms and flies.
Because IGF-1 is a key driver of aging. It promotes synthesis and inhibits protein degradation.
But this is where the complexity of mammalian aging just hits you in the face.
It really does. Because reducing IGF-1 might extend cellular lifespan. But in mammals, you need some level of IGF-1 to preserve skeletal muscle, strength, and bone density. Extreme reduction leads to frailty.
So it feels like a zero-sum game. If you slow aging by inhibiting IGF-1, but you lose all your muscle mass, have you really improved healthy longevity?
That's the challenge, finding the exact balance, or maybe developing drugs that only target the detrimental parts of its signaling.
And there's another subtle mechanism here. The ribosomes, the protein-making machines, they start to stutter in aging cells.
They do. These ribosomal translation pauses increase in frequency. This overloads the quality control systems and dramatically exacerbates protein aggregation.
And that stuttering ribosome is the upstream failure that leads us directly to the 10th hallmark, compromise of autophagy.
Autophagy, which literally means self-eating, is this highly conserved process that uses lysosomes to degrade and recycle cellular components. Misfolded proteins, defective organelles, you name it.
And this recycling system demonstrably decreases its activity with age, across all species and all tissues.
And when the recycling system breaks down, the garbage just piles up. This compromise is tightly linked to toxic accumulation, including metals. Senescent cells accumulate significantly higher levels of transition metals, like iron and copper.
Let's break down the iron link. The accumulation is due to defective autophagic degradation of ferritin. That's the protein that stores iron.
The iron can't be recycled or disposed of properly, and this process is linked directly to a specific type of programmed cell death called ferroptosis.
And it's a similar story for copper.
Very similar. Its accumulation is blocked because of a reduction in the copper exporter, which inhibits the whole degradation pathway. So these essential elements become toxic waste because the cell can't clean house anymore.
This brings us naturally to the energy source itself, mitochondrial dysfunction, the powerhouse problem.
Aging mitochondria are highly susceptible to changes in their shape, a reduction in their numbers, and just reduced function, mostly due to that accumulated ROS damage we talked about.
But this failure is paradoxical because while ROS causes damage, a certain level of stress is actually necessary for adaptation.
Exactly. This is where we introduce the concept of the redox stress signaling threshold, or RST. This is the concentration of ROS below which the stress is actually beneficial. It's a phenomenon called mitohormesis.
Wait, so you're saying a little bit of internal stress, a mild burn, is actually good for the powerhouse?
It is, like lifting slightly too much weight to build muscle or starving for a short period.
So things like intermittent starvation or regular intense exercise, they can increase that RST.
And by doing so, they can delay aging, at least in model organisms like C. elegans. It shows that a mild dose of stress can be protective by forcing the cell's maintenance systems to upregulate their efficiency.
The other key factor here is the energy currency, NAD plus off.
Right. Age-dependent decreases in NAD+ levels are seen in numerous pathologies. And NAD+ is crucial for hundreds of metabolic reactions, including DNA repair.
So replenishing NAD plus precursors, like nicotinamide riboside, has been shown in animal models to promote DNA repair and enhance mitophagy, the specific removal of damaged mitochondria.
It has. And critically, when mitochondrial dysfunction gets severe enough, leading to ATP depletion and energy failure, it can itself become a driver for senescence.
A process they've termed MIDAS, or mitochondrial dysfunction-associated senescence.
So the decline of the powerhouse contributes directly to the emergence of what you called the zombie cells.
Okay, let's move up to that cellular level and talk about them. Cellular senescence. These are the zombie cells because they stop proliferating, but they refuse to die.
And they're not just inert. They are metabolically active, but functionally destructive.
We can categorize this state into two main causes: replicative senescence, which is driven by that telomere shortening we discussed,
and stress-induced premature senescence, which is caused by overwhelming DNA damage, oxidative stress, or that mitochondrial failure. Both pathways lead to cell cycle arrest via those gatekeeper proteins, p53, p21, and p16-inc4, ARB.
But the main problem with senescent cells isn't their inability to divide, it's what they secrete.
It's their chemical output. The SASP, the senescence-associated secretory phenotype. They secrete this complex mix of inflammatory cytokines, growth factors, and chemokines.
And this SSP acts as a kind of local broadcast signal. It fundamentally alters the microenvironment.
It induces chronic low-grade inflammation, what we often call immune aging, and it can even cause passive senescence in healthy neighboring cells. It's like an infectious state of decline spreading through the tissue.
This concept just emphasizes the profound power of systemic communication. The state of the local environment is so important.
It really is.
Which brings us back to that truly remarkable anecdote from the research. We have evidence that systemic communication can also be positive.
The young cerebrospinal fluid experiment.
Exactly. Researchers showed that a systemic infusion of young cerebrospinal fluid, or CSF, actually improved memory function in aged brains.
That is a huge aha moment. It means that just diluting or adjusting the systemic environment with factors from a younger, healthier system can have immediate, quantifiable benefits.
In a complex organ like the brain.
The mechanism was shown to be primarily mediated by the proliferation of oligodendrocyte progenitor cells, the cells that make myelin.
So the young fluid prompted the growth and repair of these crucial support cells in the aged brain. It shows that the environment outside the cell carries these instructional signals that are potent enough to reverse some aspects of functional aging.
Which suggests future therapies might not just be single drugs, but complex modulations of our internal fluids.
The final cellular hallmark is stem cell exhaustion. Stem cells are the regenerative core of our tissues, responsible for repair and maintenance.
And with age, they lose their capacity for self-renewal and their ability to differentiate into specialized cell types.
And this exhaustion links directly back to those maintenance systems we covered.
It does. Autophagy, the ability to clean house, is absolutely necessary for stem cell function. Impaired autophagy in bone marrow stem cells, for example, accelerates aging and severely compromises their ability to differentiate.
If the stem cell can't purge its damaged parts, it can't regenerate tissue effectively.
And there's a clear, visible marker of this decline. Hematopoietic stem cells, the blood-forming ones, they actually get bigger during aging.
They enlarge.
They do. And this increased size likely corresponds to structural disorganization and reduced function.
Okay, so we've mapped the hallmarks. Let's transition now from the general why of aging to the specific, actionable how.
Right. We're going to show you how these 10 hallmarks don't act in isolation, but converge to drive the most prevalent age-related diseases.
And this link is the core promise of anti-aging research. If you can intercept these fundamental hallmarks, you can potentially prevent or treat complex pathologies across multiple systems.
Let's start with Alzheimer's disease, a catastrophic neurological failure defined by that dual pathology of ECI amyloid beta deposition.
Forming the senile plaques.
And the hyperphosphorylation of tau proteins, forming neurofibrillary tangles.
And the hallmarks are front and center in both of these mechanisms.
They are. DNA damage accumulation, exceeding the neuron's limited repair capacity, is a key driver. But the ill plaques themselves are toxic not just by taking up space, but by directly impairing the mitochondria.
So our oligomers, the early clumps, they don't just block synaptic plasticity.
No, they physically surround and impair the mitochondria, leading to that fatal release of reactive oxygen species we talked about.
So the decline of the energy system is central. Impaired mitophagy, the specific mechanism for cleaning out damaged mitochondria that triggers both out and tau accumulation.
And leads to compounding oxidative damage and cognitive deficits. When the garbage disposal for the mitochondria fails, the neuron just runs out of clean energy sources and dies.
And we can't forget the zombie cells. Glial senescence, especially in astrocytes, is rampant in the Alzheimer's brain.
These senescent astrocytes stop doing their normal job like clearing neurotransmitters, and they start secreting those detrimental SASP factors.
Which contributes directly to blood-brain barrier dysfunction, making the brain vulnerable, and it just fuels general neuroinflammation.
Right. Now, if we move to Parkinson's disease, it's characterized by dopamine deficiency and the accumulation of azinuclein aggregates forming Lewy bodies.
chemically distinct from Alzheimer's, but the underlying hallmark is often chronic inflammation.
We see a quantifiable increase in senescent cells in Parkinson's patients, which raises the production of pro-inflammatory cytokines like IL-6.
And the alpha-synuclein protein itself, when it aggregates, it directly interacts with the innate immune system.
It does. It increases IL-1 secretion by engaging toll-like receptor 2 and activating this massive inflammatory platform called the NLRP3 inflammasome. So it's a loss of proteostasis leading directly to chronic inflammatory signaling.
Let's switch systems to cardiovascular and vascular diseases. The heart, which is constantly working, is highly susceptible to aging.
Which makes cardiac aging the #1 risk factor for heart failure.
And here, cellular senescence plays that infectious role we mentioned. Senescent cardiomyocytes, the heart muscle cells, they promote senescence in their neighbors through that paracrine SACP signaling.
It's one failing cell poisoning its environment, which leads to widespread tissue stiffness and scarring, or cardiac fibrosis.
And this creates a vicious cycle rooted in mitochondrial failure and oxidative stress.
Increased ROS leads to irreversible damage, which triggers senescence, which disrupts mitochondrial integrity and fuels even more oxidative damage. It just accelerates the decline.
If heart failure is the pump failing, atherosclerosis is the plumbing problem. Vascular aging, or the stiffening of the arteries.
It's one of the leading causes of morbidity in old age. Senescent endothelial cells, the inner lining of the blood vessels, they lose their ability to regulate vasodilation.
They stop producing enough nitric oxide.
Right, and they increase production of a potent vasoconstrictor called endothelin-1. This shifts the balance toward chronic constriction, hypertension, and inflammation.
And it's not just the inner lining. The vascular smooth muscle cells, the VSMCs, are also critical.
Their senescence is driven by telomere shortening, DNA damage, and epigenetic changes. And these senescent VSMCs are highly inflammatory. They secrete cytokines that recruit immune cells, accelerating plaque growth and increasing the risk of rupture.
The event that causes most heart attacks and strokes.
Exactly.
And what about metabolic diseases? Type 2 diabetes is a perfect example of hallmark convergence.
It really is. The core issue is an increased number of senescent pancreatic bone cells. They have shortened telomeres, which leads to impaired insulin secretion and this state of glucolipotoxicity.
Cellular stress from a massive overload of free fatty acids and glucose.
And the hallmark link here is inflammation and SASP right there in the pancreas. The circulating fats and glucose activate the innate immune system,
which stimulates the release of massive pro-inflammatory factors like IL-1 and IL-6.
And chemokines that act like chemical flares, promoting the rapid infiltration of pro-inflammatory macrophages into the pancreatic islets, which then destroy the insulin-producing capacity.
And the SASP from those distressed dead cells then promotes senescence in their healthy neighbors,
leading to widespread dysfunction and worsening systemic insulin resistance.
Finally, let's turn to the liver. Non-alcoholic fatty liver disease, or NAFLD, and its severe form, NASH.
This is a pathology rooted in metabolic imbalance. The liver is failing to process fat effectively. It's creating too much fat and not burning enough.
And NFLD is essentially a perfect storm of hallmark failures.
It is. You have mitochondrial dysfunction, reducing fat burning and increasing ROS. You have loss of proteostasis, appearing as chronic endoplasmic reticulum stress.
And you have compromised autophagy in the form of impaired lipophagy, the specific process for degrading lipid droplets.
And of course, cellular senescence markers correlate directly with disease progression in the liver cells.
And to make matters worse, this is all exacerbated by the gut-liver axis.
Dysbiosis, or an imbalance in the gut microbiota, impairs intestinal permeability. The gut starts to leak.
And bacterial products travel directly to the liver, where they activate the innate immune system and trigger yet another layer of inflammation.
It's a cascading failure, starting with nutrient-sensing imbalance and ending with systemic inflammation in the liver.
So if the problem is this network of 10 intertwined failures, then the solutions must be targeted, precise, and often synergistic.
We have to move beyond just treating symptoms to addressing these foundational mechanisms. Let's look at the interventions, starting with the least invasive ones.
Right. We'll explore both non-drug lifestyle therapies and targeted drug treatments.
Caloric restriction remains the foundational concept in longevity, though pure CR is difficult for humans.
And sometimes dangerous.
But nutritional approaches based on its principles show enormous promise. The Mediterranean diet, for example, is rich in polyphenols and olive oil, and it significantly reduces risk for AD, atherosclerosis, and cardiovascular disease.
And its effectiveness is partly mechanistic, right? It activates SRT1 and reduces inflammation.
It does. And specific adjustments, like ketogenic diets or omega-3 fatty acids, have also shown benefits for cognitive function.
But here's the critical caveat that often gets overlooked.
Caloric restriction, while broadly beneficial in many animal models, can also have extensive and sometimes unfavorable influences on certain diseases.
So a universal extreme diet recommendation for all older individuals is just not scientifically responsible.
It's not. Beyond diet, physical exercise remains a powerful, broad-spectrum intervention. It prevents AD and heart failure progression, partly by inducing that mitohormesis and enhancing cellular cleanup.
And then we have more radical concepts like microbiota transplantation.
Fecal microbial transplantation, or FMT, is being explored for conditions like heart failure and type 2 diabetes, based on the principle of repairing gut dysbiosis to reduce inflammation.
But the clinical results are still inconsistent and controversial. It's not a simple fix.
Not yet. Since cellular senescence is such a common denominator, it's become the most targeted area of drug development. This leads to two distinct strategies.
First, senolytics. Think of these as the assassins.
They're designed to eliminate the senescent cells entirely by targeting the anti-apoptotic pathways, the survival mechanisms that these zombie cells use to avoid death.
Key examples are the combination of dasatinib and quercetin.
That combination has been shown to reduce aging markers, improve vascular function, and eliminate senescent cells in models of type 2 diabetes.
And another one, navitoclax, has improved heart function and eliminated senescent cartilage cells in osteoarthritis models.
But we have to address the toxicity challenge. ABT263, a highly potent senolytic, improves diabetes outcomes, but is also toxic to platelets.
It causes transient thrombocytopenia.
This is the high-stakes trade-off. Are we just swapping chronic inflammation for a new, acute problem of systemic toxicity? Developing highly selective senolytics remains the biggest hurdle.
The second strategy is xenomorphics. If senolytics are assassins, xenomorphics are diplomats.
That's a great way to put it. They don't kill the senescent cells. They just suppress their inflammatory output, specifically inhibiting the damaging SASP.
And metformin is the most famous example here.
It's widely used for diabetes, but also affects longevity through AMPK activation. It inhibits senescence in blood vessels and pancreatic cells by quieting that inflammatory output.
But again, the paradoxes continue. High doses have been shown to reduce mouse lifespan, and its human use can cause abdominal issues and vitamin deficiencies.
The precise dosing and mechanism for anti-aging purposes are still complex and debated.
Let's move to other key molecular targets, like NAD+ supplementation. We established NAD+ levels decline dramatically with age.
And this decline is implicated in everything from DNA, repair failure, to mitochondrial dysfunction.
Replenishing NAD+ precursors like nicotinamide riboside or NR has shown promise in animal models.
It alleviates fatty liver disease and improves mitochondrial function in mice, but here we encounter another significant clinical paradox.
The failure to translate from mice to men.
Exactly. Clinical trials giving NR to healthy obese men showed no significant improvement in insulin sensitivity, mitochondrial respiration, or inflammatory markers.
So just supplementing a precursor alone may not be enough to override complex metabolic disorders in humans.
The human metabolic environment is clearly more complex than we had hoped.
We can also look to traditional Chinese medicine, which often offers alternative therapeutic avenues.
For example, cycloostragenol from the herb astragalus is the only telomerase activator discovered to date that can delay telomere shortening. It's an intervention that directly addresses the time clock.
And it also ameliorates cardiac insufficiency and heart failure by increasing autophagy, so it's hitting multiple targets.
Another powerful compound is berberine. It has significant hypoglycemic effects in diabetes, partly by modulating the gut microbiota, and it treats atherosclerosis by regulating cardiovascular risk factors.
And finally, we have advanced methods for tissue and cell repair. Stem cell transplantation is being used for osteoporosis and COPD.
And a highly promising targeted therapy is using iPSC-derived RQE cell transplantation for dry, age-related macular degeneration.
Replacing the critical cells for vision.
And it's already showing improved visual acuity in patients, representing a genuine functional cell replacement success story.
Now, cancer presents the ultimate complication in aging research because cellular senescence has this deeply dual and contradictory role.
On one hand, it acts as a critical barrier to tumor genesis.
That's its initial tumor-suppressing role. It forces a stable growth arrest, stopping potentially malignant cells from proliferating early on.
And it could even use the SASP initially to recruit immune cells to clear the incipient tumor. It calls the cops.
But if those senescent cells persist, the role flips completely. They become powerful tumor promoters.
The long-term SASP creates an immunosuppressive, chronic inflammatory microenvironment. Factors like IL-6 and VEGF actively drive tumor growth, angiogenesis, and metastasis.
The senescent cells create fertile ground for aggressive tumors to take hold.
So the ideal therapeutic strategy for cancer has to be a sophisticated two-step process.
You have to use both sides of the coin.
Exactly. The first step is senescence-inducing therapy, using chemo or other drugs to halt the growth of tumor cells. The crucial second step is senolytic therapy immediately after, to clear those potentially harmful senescent cells you just created.
Preventing them from ever becoming tumor promoters.
That's the idea.
So, we have covered a monumental amount of information, mapping this entire landscape of decline. If we synthesize the key takeaways, the most crucial realization is that the 10 hallmarks of aging are not independent events.
They're an interconnected catastrophic web of failures: mitochondrial dysfunction, defective autophagy, loss of proteostasis, and cellular senescence, with its damaging SASP. They emerge as common denominators driving chronic disease across all systems.
And this leads us directly to the central challenge for intervention. Anti-aging drugs face significant hurdles, whether it's the known toxicity of senolytics or the paradoxical and inconsistent results we've seen in human trials for compounds like metformin and NAD plus precursors.
The future success of this field is predicated on moving beyond single targets. We have to develop synergistic combinations of treatments tailored for individual patients, which is just one of the greatest scientific challenges of our time.
So what does all this mean for the future of medicine and for you, the listener?
I think this raises the most important question. The focus of geriatric medicine is shifting, and shifting fundamentally. We are moving away from merely treating symptoms after a disease has already started,
The classic reactive medical model.
and moving toward targeting the underlying aging-related chronic disease risk factors for pre-intervention and prevention.
This means integrating new technologies, genomics, AI, to analyze those molecular markers, like the epigenetic clocks we discussed, to get a comprehensive assessment of a person's true biological age.
The goal is no longer just extending lifespan, but extending health span by treating aging itself as the primary condition to be managed.
The greatest shift in public health may be toward these personalized molecular assessments.
Identifying which of the 10 hallmarks are failing fastest in an individual and intervening years or even decades before the symptoms of Alzheimer's or heart failure ever manifest, that is the true promise of healthy longevity.
Shifting our entire healthcare focus from reaction to profound prevention. A truly transformative vision for the future. Thank you for joining us on The Deep Dive.